put your questions in the chat, and we will get those relayed to our speaker. So without further ado, the next workshop will be given by Lambda Moses, and it's on exploratory spatial data analyses from single molecules to multiple samples.
All right, so yeah, if you you probably already have workshop.bioconductor.org open from the previous workshop. So once you get to workshop.bioconductor.org, you can navigate here, like on the left, it's this one. Yeah, click Run Tool. Yeah, so while it's might take a while to pull the Docker image, so like I can. Yeah, so a rendered version of this workshop can be accessed at, here, but it's linked to on here. Yeah, it's open. What bad gateway? Okay, here we go. Yeah. So is that large enough? Right. So is that large enough? Okay, yeah. So here in the Docker container here, you actually have links to the rendered version of the workshop in the README. No, it's not the README. It's in the workshop.rnd in the vignette. Yeah, so if you want to, so this is what the rendered version of the workshop looks like. Yeah, so basically, initially I would do some intro and yeah, we have a lot to cover here, and yeah, so we can get to the intro. Hi, my name is Lambda. So I was a graduate student and a postdoc with Neuropactor at Caltech, but just last week I just moved to Columbia in New York to start my new postdoc with Bianca Dumitrescu. And yeah, today actually pretty much I would talk about the updates in SpatialFeatureExperiment and the Voyager packages. Yeah, so I will begin with an introduction to the a little bit of background about spatial transcriptomics and the philosophy of my packages, and then I will get to the so the new features. So mostly about read functions and about the images and spatial aggregation. Yeah, so if you have been to my workshop before, like last year, I often say like from geospatial to spatialomics, because the geospatial tradition really has inspired a lot of ideas in my packages. And here in so here's the 2020 census data from a few states showing the demographics of and however problematic these categories are. Yeah, you can see here that it gives you some information, like California tends to be more diverse compared to some other states. But meanwhile, this data has, it's missing a lot of information. And if we disaggregate those bar charts and map that into space, like here shows much of LA County and where each demographic group is located. Just having spatial information can make us ask many interesting questions, like so for instance, which groups are located where, if they, so yes, we do have spatial regions that are dominated by certain groups, and also like who tends to co-localize with whom, and so on and so forth. And actually spatial distribution of not only demographics, but also like infrastructure and so on is very important to how LA functions, both for good and for bad. And in histological space, it's spatial distribution of gene expression in different cell types is also very important to the proper function of the tissue. And again, just like in geographical space, by having the spatial information, we can also ask many questions like which genes have expression pattern that is spatially structured, and which genes co-express in space, what are the archetypal expression patterns, and what are the spatial regions, and so on. So, that's why, we may have heard of a cliche that says the whole's greater than the sum of the parts, and so that is because of another cliche, location, location, location. So right now, we already have technologies that can profile the spatial distribution of gene expression across the whole genome. Well, often, maybe from a few hundred genes to thousands of genes to transcriptome wide. And here so this plot shows that the drastic growth in the number of new publications on spatial transcriptomics over time, and that indicates a drastic rise in the research and interest in this field. So right now, spatial transcriptomics is already mainstream, and some of the technologies are already commercialized and used by hundreds of institutions around the world, although in practice, pretty much mostly in the high income regions of the world. Just a quick crash course on the technologies used to collect spatial transcriptomics data. The most popular, largely there are two categories. There are those based on next generation sequencing, like Visium, already used by over 300, now probably more institutions. How it works is that you have a tissue section, and you can usually stain that with H&E or some sort of fluorescent stain, immunofluorescence, and you mount this tissue section onto a special microscope slide, and that slide has a hexagonal array of spots that have barcodes whose spatial locations are known, and it also has a poly-T sequence to capture polyadenylated mRNA molecules in situ from the tissue, and so from the capture we can basically sequence the transcripts to quantify their expression at each spot and map. And we can map that to locations in tissue. But this array, actually the spots, they do not have single cell resolution. Well, at least the classic, the older version of Visium. There can be several different cells in the same spot, but this is transcriptome wide. Another major type, so like MERFISH, molecular cartography, CosMx, and Xenium, those actually also in situ sequencing. So those are imaging based, and so basically we in these technologies we can visualize each mRNA molecule in each cell as a discrete puncta in space, and we can actually relate each spot to the gene it comes from. And then by counting the number of mRNA puncta assigned to each gene and in each cell, we can quantify how much each gene is expressed in each cell. So these technologies not only have single cell resolution, but also single molecule resolution. And however, usually these are not transcriptome wide, and so usually people would quantify a few hundred genes per dataset. But this is the number of genes is also increasing with the new version of Xenium that has a 5K gene panel. Yeah, so that's a very short intro to data collection for spatial transcriptomics. And so once we have the data, we need to analyze the data. And broadly speaking, the spatial transcriptomics data analysis can be divided into upstream and downstream. For upstream analysis, the method will take in the raw data from which would depend on technology, and then process the raw data into a more usable form. Into a more usable format for downstream analysis, and in downstream analysis we will get to the biological inferences. So for my packages, SpatialFeatureExperiment as a data structure, as a way for bookkeeping, it's So it's more like upstream, whereas Voyager for exploratory spatial data analysis, it's more downstream, and both packages are on Bioconductor, and you can install them with BioC Manager. So now we get to spatial feature experiment. Well, you can basically think of a spatial feature experiment object like a layered map. So like here, here I will introduce the major types of spatial data that in geography, which whose infrastructure I have borrowed to spatialomics in spatial feature experiment. So in this map showing much of LA County, we see the polygons here. These are boundaries of cities in LA County. These polygons are defined by the longitude and latitude, the coordinates of the vertices, and actually there are lengthy legal documents just writing out the coordinates of each vertex. Meanwhile, at each of these polygons, we can have data associated with the geometry. So like here, it's this data set shows the number of homeless people in each neighborhood in LA City and in the smaller cities in LA County. There are other kinds of data that can be described by the coordinates of the vertices. For instance, here so just now showed like a polygon geometry described by the vertices, and here it's rivers in LA County, and the rivers are line stream geometries, and they can also be described by the vertices. And how many vertices would depend on what resolution of the map you need. Meanwhile, here we also have polygon geometries of the flood basins and the reservoirs, and the point geometries also like described by their longitude and latitude coordinates for the location of dams. So just now I said the word vertex a lot. So basically when you use the coordinates of the vertex to describe a geometry in space, that's called vector geometry. So that's one of the major types of spatial data in geography, but there's another major type called raster. Like shown here, basically a raster data is a array. It's a image where each pixel in the array has data, has values. But it's more general than your average photograph because the values at each pixel, so actually the image can have any number of channels, and the values at each pixel can also be like categorical, as shown here. This is land use in LA County, and in addition, unlike a photograph, the raster data must have a spatial extent. It has to be registered to the spatial coordinate system. So here in say this image, we have to know the longitude and latitude where this image begins and ends, so we know where it's located in space. And so because of that, this is actually aligned to the to the same spatial coordinate system as the vector geometries, like where City Hall is located for each city. So a more familiar example of raster data will be the satellite image like from Google Maps. And here it's the raster image is aligned to in the same spatial coordinate system as the vector geometry of the freeways. In R for vector data, so actually in geography, simple features is a standard way to represent to represent vector geometry data, and in R, the infrastructure for simple features is provided by the SF package, but it's really an R interface to a lot of C++ libraries like Geos, GDAL, Proj, and S2 actually. And it's C++ libraries that performs the geometric operations on the data. So in R, basically, it's SF data is it's actually a data frame, and so I mentioned that each geometry we can have data. So like the number of homeless people per neighborhood or per city. So those will go into the columns of the data frame, but there's a special column for the geometries. So the special thing about this is that we can, it's the geometric operations, and so that's where we can find like whether two geometries intersect and find the geometry of intersections and so on. So for for spatial omics, the geometries can be cell segmentation polygons, can be the location of the mRNA molecule. So with say geometric operations, we can find which mRNA molecules in which cell, or like which cells in which cell intersects with which Visium spot. And so with this, we can quantify we can see like how many mRNA molecules there are in each cell and so on. For raster, actually there are several different packages in R that can work with raster, like Terra and stars. But here I mostly use Terra for raster. And so Terra, I think it's very it comes from the geospatial tradition. I think it's very well written in C++, and is the cool part about Terra is that it actually also has some functionalities for vector geometry, so that way we can use the vector geometries to extract data from raster. And I'll show an example of this in the coding part of the workshop. So spatial feature experiment, we can think of it like a layered map. So kind of like this is a Visium dataset from the mouse skeletal muscle, and so like we can have a raster layer for the H&E image, and we can have vector layers for the polygons of the Visium spots and for the myofibers, and those geometries can have data like gene expression for. Or like some metadata like total count library size for the Visium spots, and like morphological metrics like area, like eccentricity for the myofibers. And yeah, another example, like it's the raster layer will be the DAPI image for fluorescence of fluorescent data. That's MERFISH, and we can have vector layers for the cell segmentation polygons and for the transcript spots. So like I'm pretty sure like a lot of you here have used tidyverse, and so yeah, in tidyverse if you've used it for long enough, like you may have taken the tidy data frame for granted, but tidy data frame is actually very instrumental to non-spatial exploratory data analysis. However, like not all data, so not everything is best represented by a tidy data frame. So yeah, an example of that will be single-cell RNA-seq data. And spatial transcriptomics has inherited a lot from the single-cell tradition, the non-spatial single-cell tradition, where we would dissociate the cells from tissue, losing the spatial information. And for actually a lot of the methods, like for high-dimensional data analysis, still relevant for spatial. In the Bioconductor ecosystem, the single-cell experiment is used to represent and operate on single-cell RNA-seq data. Like it centers on a gene count matrix with genes in rows and cells in columns, and we can have metadata associated with the genes and the cells, and we can have the projection of each cell into a lower dimensional space. So SFE, because we're here, like we're spatial transcriptomics, we're actually focusing on gene expression. So another perspective to think of spatial feature experiment, or SFE, is it's actually a spatial extension of single cell experiment. And so again, we have the gene count matrix in the middle here, but the spatial aspects are added via, so for vector geometries, it's from SF, and for raster, it's from Terra. And the SFE is actually not the first attempt for spatial extension of SCE. There's already an older class called spatial experiment, which SFE also builds upon. Spatial experiments, where SPE has introduced spatial coordinates field for the X and Y and sometimes Z centroid coordinates of the cell or Visium spot, and it has added the image data field for the images, although it's only for visualization. And SFE has actually inherited some of the getters and setters from spatial experiment. So now I would get to the different parts of the different fields in the SFE object, and here to introduce those, we'll use the code here in the workshop, and I will go back and forth between the code here in the workshop and the slides. So first we will load the packages. Yeah, so you can go to the documentation website for SpatialFeatureExperiment and Voyager for more details about some of the topics brought up in this workshop. But actually, some of the materials here is new, and it's not on the documentation website yet, and I'll add them later. So number one, how do we construct a spatial feature experiment object? Yeah, we can do it from scratch. Just it's mostly similar to single cell experiment. So here we used just random number generator to create a gene count matrix, and we also created the spatial coordinates. The spatial coordinates can be supplied through call data, and we call the spatial feature experiment constructor to construct a spatial feature experiment object from scratch. But most of the time, yeah, I have observed from the Museum of Spatial Transcriptomics dataset, a database that commercialization of a technology will really make the technology more popular, like a Visium, also MERFISH, it got more popular after it got commercialized. So probably most of the time the user will use one of those commercialized technologies, and each of those will have its own data preprocessing pipeline, and which will give you a output directory with a standard structure. So Visium is the most popular one, most popular data collection technology, and the Space Ranger is the official data preprocessing pipeline. And so here, SFE package comes with a toy example, Space Ranger output for Visium. It's the old-fashioned Visium. So we have the gene count matrix and the spatial information, which includes the images and the spatial resolution of the images and so on. Well, but here because the example data sets we're using in this workshop are from imaging-based technologies, that's where most of the updated in my packages are about. I'll just gloss over the Visium part. So basically, you use the read 10x Visium SFE function to read the output from Space Ranger. So this will read the output as a spatial feature experiment object. And this is what the show method of SFE will look like. It will give you the dimensions. So here in this toy example, we only have 5 genes and 25 Visium spots. Yeah, it shows a summary of the raw data. So that's the gene metadata and call data. So that's the metadata for the Visium spots. And but in addition, it shows a summary of the images. And image data and what geometries there are, and here we have spot poly, so that's the polygon for the Visium spots. Okay, so now we'll get to the data sets that will be further used later in the workshop. So the first one's Visium MERFISH. So there's the package SFE data has example data sets, actually mostly used for demonstration and unit testing purposes. And here from SFE data, we download an example output directory for VisGen MERFISH. So the first time you run this code chunk, like with VisGen output, actually whenever you run the first time you run anything that uses experiment hub, so it may appear like the code chunk in our Markdown notebook is stalled, but in this case, you just open the R console. There's a prompt this experiment hub directory doesn't exist. Create a directory, yes. So this will download the VisGen output directory. Yeah, this is the structure of it. So we have actually even different versions of VisGen would have a slightly different output structure, and so the read function for VisGen can accommodate different versions of the output. So here we can have one kind of so that's one type of cell segmentation So here, like each field of view will have the vertices of the cell segmentation polygons in the HDF5 file, but there's another version available in some outputs. So cell boundaries.parquet. So that's from CellPose. And we have the gene count matrix, the cell metadata, and so detected transcripts.csv. So that has the X and Y coordinates of all the transcript spots detected in this data set. And for images, for at least the newer versions, we have 3 different kinds of cell membrane stains. So that will really help with cell segmentation. We also have DAPI for staining for nuclei and poly-T for staining for all the polyadenylated mRNAs. So here we use the read visgen function to so here that's just the directory, the top level directory of the output shown here. Yeah, you can select which z-plane to read. For MERFISH usually has 7, but for cell segmentation they're all the same. And there's the add molecules argument. It's optional, but if you set it to true, the function will process this file, the detect transcripts file, so after processing, the transcripts will be added to the SFE object. Yes. Can you stand over there so the online audiences can hear it?
Can you hear me?
Okay, yes.
Excellent. So first question, as you've pointed out, these technologies are quite new, and output structures, as you show here, this tree, as well as format changes frequently. How do you deal with that for your functions?
So I think actually it depends on technology. Like for Visium, it's pretty stable. So, but for VisGen, like it has different versions, and so internally the read VisGen function will try to determine like which version, and it will like it tries to deal with like different scenarios.
Okay. So how often do you end up updating your functions?
I'll also update the function.
Yeah. And then the other thing on the add molecules. So I guess for these small example data sets, no problem to read them into memory, but for real data sets, these are huge data frames with millions of molecules. Are there any concepts of like out of memory, any database backend, anything like that?
Actually, like it's in the text here. It's still like a kind of in the very beginning of the development. Actually, like for one of the example data sets, like the detected transcripts.csv files, like 30 gigabytes, and then like I will have to use a server to process them. But after, so I will get to that here. Actually, for VisGen MERFISH, the cell boundaries are already in GeoParquet format. So it's like it's not just plain vertex coordinates, but those are simple feature objects already. So like I don't have to process this one. The GeoParquet files are much smaller compared to the original CSV. So like after I process them, so like the read function will process the detected transcripts.csv file and into like GeoParquet and read into disk. And here we can see that even for the toy data set, the GeoParquet file is much smaller compared to the original CSV file. So next time, actually, for that data set with like a 30 gigabyte detected transcript.csv, like after processing the GeoParquet file is much smaller, so I have no trouble loading that into my laptop's memory. So like here I already wrote like a Parquet, what Parquet is. It's actually also like for GDAL. I think it's also in the text. GDAL is like kind of a uniform user interface to a lot of like different drivers to read different spatial file formats, and it actually has like a SQL, like its flavor of SQL. So like so if you have a newer version of GDAL, like it requires like at least 3.5.0. So that will support a Parquet driver. So that way, once you already have the GeoParquet file for the transcript spots, you can use. So there's a function in the SFE package called read select transcript. So there you can. I think most of the time probably the use case for the transcript spots will be like visualization. Probably most of the time. So with the read select transcript, you can specify like just a small number of genes and then GDAL behind the scene, GDAL will just only read the transcript spots from those genes instead of everything. And that's also like called behind the scene by the plotting functions that will plot the that will plot the transcript spots. But again, like that would require like a newer version of GDAL. But also like I have plans to I haven't implemented that yet. For like right now, the cell segmentations are also low into memory. So far, like even for datasets with hundreds of thousands of cells, I haven't really gotten to memory issues even on my laptop. But in case you have like multiple samples, like millions of cells, I might not be able to do that on my laptop. But so actually, like there are in geospatial field, there are already like a lot of large data sets, and there are already infrastructure that can allow us to perform the geometric operations on like out of memory. So for instance, there's the Apache Sedona package, and it's actually on CRAN. It's also like multilingual, and there's also DuckDB. So I might use those in the future to do the geometric operations out of memory.
Yeah, I think we will need that going forward. Data is only going to get bigger. I think that will be great.
Yeah.
A question online from Terry wanted to know if you could also read in the Visium HD data.
We're working on that, so it's already in the develop branch. Although I do have a plan. Actually, it's not just me, but also Alec Hosseinoff from DKFZ. We do have a plan to make a example data set for unit testing and to write a vignette for Visium HD. And also, like I have a collaborator who is trying to collect Visium HD data, so that will come to me naturally. Yeah, so basically, the code is there. It doesn't require a lot of changes from like old-fashioned Visium HD, but I haven't written unit tests yet. Yeah. All right, so next, 10x Xenium is another one of the popular, it's getting more popular commercial like imaging-based technologies. And again, so there is a subset of a real data set from 10x website provided by the SFE data package. So actually Xenium, they're already, at least the pre-processing software, the Xenium onboarding analysis already has 2 versions. They're already in release. For here I'm reading one from V2. Yeah, so this is what the output structure looks like. So we have cell boundaries, and actually, so that's for cell segmentation polygons, and we also have nucleus segmentation polygons. So for XOA V2, the unlike V1, the segmentations are greatly improved, and also V2 will allow multiple nuclei in each cell.
Beneath, do you recommend running cell segmentation on the H&E and DAPI pre-Visium to then align to the image for this type of analysis?
Yeah, you can do that. Although it's not like Space Ranger doesn't do it, but you have H&E, but so you can do it. You just have to do it on your own. And actually, I have done it myself for one of the Visium example data sets in SFE data. Yeah. So back to Xenium. So we have cell and nuclei segmentation, and but here, although we have the Parquet file, it's actually not GeoParquet, and internally it's just the X and Y coordinates of the polygons, and we have the gene count matrix and the cell metadata, and also we have the transcript spots. But here the Parquet file is not GeoParquet, so as a result, the read Xenium function would process those and convert those into GeoParquet. And for images, for XOA V2, there are 4 channels, and it's in the OME-TIFF format. So it's from the open microscopy project. So here, here I would load the data into R. Oh, here it's like installing our BioFormats. Yeah, so while it's installing our BioFormats, so basically BioFormats is a Java library that can read a lot of different microscopy file formats. So model will be OME-TIFF, and which Xenium uses, and but also like some commercial like proprietary formats from like a CZI, like ND2 and so on. Also ZAR. Okay, so here it's range R. Yeah, you see the messages about processing the coordinates into GeoParquet and writing that to disk. So here this is a data set. We have almost 400 features and over 6,000 cells. And we have a summary of the col geometries and row geometries and so on. So here in the directory, we see that we have the sf.parquet files written in, by default into the same output directory. So next time we read the data, it will read Xenium will read the GeoParquet file, which will be faster. Yeah, and txspots.parquet will be the GeoParquet for transcript spots. So far, SFE also implements refunctions for other technologies, in particular recos-MX. So that's for nanostream cos-MX, but because we're not actually using the dataset here, it's not shown. But meanwhile, yeah, we haven't gotten the chance to implement the refunctions for other commercial technologies, like Koya, like molecular cartography and the Curio Seeker. So Curio Seeker is the commercialized version of SlideSeek, and so if so contributions are welcome, and you can do a pull request on GitHub.
Hey, I have a question.
Yes.
What do you think about spatial experiment versus SF? If let's say we don't yet want to use the polygon boundaries and just we want to work with centroids, is there, like, I'm trying to figure out when would one want to switch from spatial experiment to this?
So basically, I'll get to more of that later in the workshop. So basically, in spatial experiment, you just have a matrix for the x, y, and sometimes z centric coordinates, but that's just a matrix. But for SFE, the centroids, you can't actually put centroids in the col geometries, and because it's SF, because it is simple features, you can perform, like you can use in say, like geometrical operations to see like which cell has a centroid within a spatial region, or like within like a spatial bin, or like a histological region, and you can also use a polygon to crop the your SFE object and so on. And you can also find spatial neighborhood graphs from those. Yeah, it's just SFE has added a lot of features to spatial experiment.
Okay, cool.
Yeah, you're welcome. Yeah, so like here, we just got to spatial experiment. There are already some spatial omics data analysis tools that already use SPE. But if you do want to use SFE for some reason, you can convert you can convert SPE to SFE. So for instance, here read 10x Visium. So that's from the spatial experiment package, and so here I loaded a toy example for Visium. And then you can call the toSpatialFeatureExperiment function to convert SPE to SFE. I think more importantly, in both single cell and spatial omics, Surat is by far the most popular package for data analysis. So Alec has contributed function that can a Seurat method for two spatial experiments. So that way we can convert Seurat objects into spatial feature experiments. So that we can do more of those geometrical operations, and we can use Voyager for exploratory spatial data analysis or ESDA. And here's an example, but so far we haven't gotten to like convert an SFE to Seurat yet. Yeah, so then so that was on constructing SFE objects, and then now we're getting to the different fields of the operating on SFE objects and on the different fields in the SFE object. So SFE actually inherits from SCE. So here's just a quick recap on SCE. So because SFE inherits from SCE, it also inherits all the getters and setters, and also like there are already like an ecosystem of single cell analysis tools for SCE, like Skater, Scran, Scuttle, and Bluster, and so on in the ecosystem. And because of the inheritance, like all those methods will still apply to SFE. So in SCE we have the gene count matrix, and the raw counts, the raw, like say UMI or the molecule counts are can be get or set with the counts getter or setter. Yeah, so here this is the Xenium data, and so this is what the raw counts matrix looks like. Remember there was the H5 file for this. So this is read as delayed matrix. So you can go to the documentation for delayed array package to read up more about it. So basically delayed matrix does not load the so here for H5 it does not load the entire thing into memory, and it really helps with analyzing larger data sets. So Aralone has used a delayed array to analyze single cell data set with over 1 million cells on his laptop. Yeah, the setters looks pretty much the same, and so for the metadata associated with the cells, we use the call data function. So it's a data frame when we have the total counts come from the Xenium preprocessing, like total counts, like the cell area, nucleus area, and so on. And raw data it's analogous to call data, but it's for gene metadata. Like here we have the ensemble ID, and we have the gene symbol, and the type for actually not all the features are for gene expression, and some of those are like negative controls, and they do not target genes, and that's used for quality control purposes. Just like in SCE, for SCE is centered on the gene count matrix, and it can be subsetted just like a matrix. And here I would do that subset just like a matrix to do a quick dirty QC to remove cells with very few counts, very few gene counts, and to remove the features that are not detected. And then here I'm just again I'm just applying the methods from SCE. It's not spatial yet. Do the data normalization, and so after doing the log normalization, the log counts getter from SCE can be used to retrieve or to set the log normalized count matrix. And then we can use run PCAs from Skater and from BioC Singular. It's already part of the SCE ecosystem. For single cell experiment, the results from dimension reduction are stored in the reduced dim field. Yeah, like so reduced dim, it's kind of like a special case for call data. So in both getter and setter for reduced dim, we put the SCE object here, and then we use the name of the dimension reduction to get or set the results. Like in this case, it's PCA. And here we get the projection of each cell into the lower dimension, like PCA space here. And because we use the name here to get or set dimension reduction results, what if you don't remember what names are present, or like which name, whether you have like lowercase or uppercase for PCA, you can use the reduced names to to get which names are available. So for the new fields in SFE, like the say call geometries, row geometries, also like call graphs and row graphs, I intentionally modeled those the getters and setters after the existing ones for SCE, so that way it will make it easier to learn. So just like reduced dims, call geometry, again, you put the SFE object here and use the name to get your set. And then here we get the cell segmentation, and we see an SF data frame with polygon geometries. And in ggplot, we can use like ggplot plus geom-SF to plot any SF data frame. And here's, yeah, this is a small subset of a real data set, and I just cropped it using the bounding box, and this is what the cell segmentations look like. But also instead, like you can, instead of like using the getter to get the geometries and directly calling ggplot to plot it, you can, there's also in the Voyager package, we can also use the plot geometry function. And in order to plot the call geometry, we populate the call geometry name, the call geometry name argument. So also like there's a light theme, which is default, and dark theme, you put dark equals true to use the dark theme if that's your personal preference. And here the plus, so this way that's from the patchwork package, which can assemble different ggplot plots into like a single plot. The dark theme basically has dark background, but also the default colors for geometries will look better in the dark theme. So here, like we have like okay, now after QC is like 5,700 something cells. It's in some places it's what if you want to zoom in, so you can use a bounding box specified by xmin xmax, and so here by showing the axes you can kind of get the coordinates of the bounding box you want to use. By populating the bbox argument in plot geometry, actually in any other Voyager plotting function that plots geometries, we can only plot what's inside this bounding box. So actually, there are many tools that like Semway browser, whose paper I reviewed, and there's also like Tissue Map 3 that can do interactive data visualization for spatial data. Honestly, like I personally don't have the expertise to write that kind of thing. I'm not very good at Shiny and or Plotly. So far the Voyager plotting functions are make static plots, but meanwhile I think static plots still have their place, like in publications. Also by supplying a vector into the call geometry name. Actually, I can on any like geometry name arguments in plot geometry, we can also plot multiple geometries simultaneously. Like here we plot both the cell and nuclei segmentations. Yeah, and we see that here. We see that some cells have like 2 nuclei, but some of them don't have nuclei. For like it could just be that this is just like a one tissue section out of a 3-dimensional tissue, and it could just be that the nucleus is not in this Z plane, or just the segmentation the segmentation algorithm didn't detect it.
I have a quick question about
yes.
the plotting functionality. So sometimes when I'm plotting these single cell or Visium data, the cell boundary or the Visium spot boundary could create a illusion, meaning it creates a shade by the color plotting on it. Do you have some specific tips on the visualization?
What do you mean by shade?
So if you go back to your the dark background plot, right? So you have a white boundary, like the inside of each cell is dark blue, and then it seems to have white boundaries of each cell, is it?
Yeah.
So the white boundary kind of give a white shades of the blue on top of it. I mean, it sometimes works, it sometimes not, but just curious if you have experienced that.
I don't have such experiences, so I don't know. Sorry.
Got it.
But you can open a GitHub issue to describe it in more detail, and I can see if it happens. Yeah, so setter is like pretty much the same, and just like for reduced dims, we supply the name if you don't remember what names are present, like you use call geometry names, and here we have centroid cell segmentation and nuclei segmentation, but also just now when it comes to SCE, you remember we used the counts and log counts getter, and so it's actually that's actually shorthand to the assay scatter and setter, and similarly here, like we have cell seg and nuke seg for cell and nuclei segmentations just based on name. So that was for yeah, so that was for the call geometries, and then next we can get to the row geometry. So those are so far the only use case I can think of is like transcript spots. However, the implementation is very general, so it really can be used for any simple feature geometry that are associated with the rows of the gene count matrix. So you can, just in case like someone thinks of like another use case.
Quick question.
Yes.
Andrani wants to know if those presentation slides will be available anywhere online.
Yes, I already uploaded that to like a Google Doc thing from the conference, but I think I'll also push it to GitHub later.
Okay. And then Beneath wants to know, how do you set your thresholds for transcripts and genes?
What do you mean by threshold? Oh, so that one. So it's actually in this case, I just removed the transcripts and I just removed the genes that are not detected. But like also like in this vignette, like it's kind of arbitrary. But I think yeah, like some more expertise about the tissue of interest could help.
Yeah. And then Beneath also wants to know, when you're reading in the Seurat data, it was pulling in just the RNA assay, and why not SC transform, or can it do both?
It's actually SC transform. Actually, you don't really have to use Seurat in order to use SC transform. I think there's a way to just use the matrix for SC transform.
And then one last comment from Tyrone. I'm not sure where you wanted to have Voyager. I guess if you're trying to handle raster image formats, it would require something like Magic rather than something like Terra for raster. And then I meant Voyager.
Lossless. Yeah, I'll get to that a bit later. So
I have a quick question.
Yes.
So when you're normalizing your data, you're using single cell kind of algorithms to normalize, right?
Yeah.
So I've been reading a lot about how those single cell normalization methods are not accurate for spatial.
Yeah, it's true. So which is why here the way I did it here is a little different from the standard single cell workflow. So like here, I did not use the total counts, but I used cell area. So that's I linked to a paper here about this problem for imaging-based technologies with like a curated gene panel. But actually, this is an open question. So like I think studies are emerging about this. Like recently, I also saw a paper about like that tries to model like the spatial autocorrelation when it comes to data normalization.
Okay, awesome. Thank you.
Yeah, so here, let me get back to this. Yeah, I'm a bit behind. Yeah, so basically for row geometries, the user interface is like pretty much the same as call geometry and reduced DIMs. So here I get the transcript spots from the Xenium, and we have multi-point geometry. So we have one multi-point for each gene, and the multi-point Z here. So actually these coordinates are three-dimensional for Xenium. So actually the SF package actually works with three-dimensional data, although when it comes to spatial, the geometric operations, it only operates in X and Y. I think it's largely because in geography, we for the most part, we live on the surface of Earth, and so X and Y, the longitude and latitude will be more relevant than altitude in most cases. But also I think for now, at least for now, for in histological space, it's fine because the vast majority of data is in thin sections. So like mostly still like X and Y, but I know that there are some data sets that have like a 3D volume, like thick section, 100 micron, like that. And there are also some with like multiple Z slices, especially in the mouse brain. those aren't many, but if they become more common, then yeah, actually we do have a plan to expand SFE to accommodate those. So again, yeah, so here the setter is pretty much the same, and for transcript spots there's a shortcut, so you don't have to like supply the name every time. To plot this, plot geometry function can also be used to plot the row geometries, the transcript spots, just to supply. So instead of call geometry name, we populate the row geometry name argument to plot the transcript spots, and everything else is the same. Like you can also use the bounding box, and the gene argument is used to specify which genes you want to plot, and you can plot all of them. So we're definitely overplotting. We see like there are those like solid black regions, and that's not good. But so to address that, one way is to so we can plot a 2D histogram to show the number of transcripts from like any gene. It can be like all gene, but you can specify like individual genes. Or it can supply a vector of gene symbols or IDs. So here the different genes are distinguished by point shape. That's because so if you want to plot cell level data, and those so that will be so the data will be we use color. And in order to make the plotting functions colorblind friendly, if I use like different palettes, it's less likely to be colorblind friendly. So here I use the point shapes, which means we can only plot like a small number of genes at a time. But when we plot like too many genes, like you get like overplotting anyway. And you can also combine the call geometry and row geometries when plotting in plot geometries. Like here I plot like cell and nucleus segmentation, and also the transcript spots for a few genes, and yeah, I can also plot the transcript spots for along with cell level data, and yeah, it's just what the dark theme looks like. And you can plot the transcript spots for different genes in separate panels along with gene expression data. So getting back to the slides, there's another field called annotation geometries. So in spatial omics datasets, there can be geometries that are clearly a part of the dataset, but they do not directly correspond to the dimensions of the gene count matrix. For example, there can be tissue segmentation, just tissue boundary, or pathologist annotated histological regions, or in Visium datasets, which do not have single cell resolution. You can, like with the H&E, with the histology, you can do cell segmentation anyway, and so, but that would not have one-to-one mapping to the Visium spots. So so those kind of geometries can go into annotation geometries. So here this Xenium dataset does not come with like annotation geometry. So I did some annotation in QPath, just to yeah, just like different sides of a blood vessel, but because the tissue 3-dimensionals are actually the same piece of tissue. So the getter and setter have the same user interface as call geometry, row geometry, and reduced dim. And here we have 2 polygons in an SF data frame. So it's yeah, we can use ggplot to plot it. So this gap will be a blood vessel, and we can also use the plot geometry function, but populate the analog geometry name argument. This can also be plotted along with other geometries like call geometry. So here I plot both cell segmentation and my own annotation from QPath for like pieces. Yeah, there's the analog geometry name, so that's the same as in call geometry names to see like what are the names of the annotation geometries that you have. So next, there's the call graph, analog graph fields, and so those in some spatial analysis methods, we need to supply a spatial neighborhood graph. So that will indicate spatially who's the neighbor of whom, and also the edge weights will be how strong their neighborhood associations are. In SFE, the main function to compute the spatial neighbor graph is find spatial neighbors, and so this is the one function ruling all for many different kinds of methods that can find the graph, and the margin argument will be, so here 2 means for column geometry, and if you put 3 will be annotation geometry, and technically there is like row geometry, although I'm not sure what the use cases are, but because internally there's a general internal function, it technically it is implemented. So here I would do a triangulation for of the cells in the Xenium dataset. So the getters and setters for call graph and anograph, and those are also the same as for call geometry. So here I put this graph into the SFE object, and I call it triangulation. And here we plot this. It's a very dense graph, and we see there's a lot of connection across the different sides of the blood vessel. Here we can also use you can also use k-nearest neighbor to find the spatial neighbor graph. And we plot it. So it's much less dense, and it does not connect across the different sides of the blood vessel. So again, just like for the geometries, here the getters and setters, you have the name here. If you don't remember, you have the call graph names. So now we have triangulation K and N. All right, so when it comes to the new functionalities, so we have image data, and the SFE has greatly enhanced the image data. Here, they're mainly so SpatialExperiment has its own image classes, but SFE has different image classes. So there are different image classes because the spatial extent is required to so that the image, just like for your satellite image, with the spatial extent, the image can be aligned to the same spatial coordinate system as the geometries. So mainly, I've used Terra for spat raster, that's for the geospatial tradition. So the example here, I use MERFISH dataset for that example because it's not OME-TIFF, and the images are read as a spat raster from Terra. So we use the get image getter to retrieve the images from SFE objects, and actually this is inherited from SpatialExperiment. We can also use image data getter. It's also from SpatialExperiment to list out the other images. And here we see, we get a summary of their class and their dimensions, and we see that here they all have all of them only have one channel. The image ID, so we want to use this to plot the images, and in case you don't remember all of them, you can use the image ID's function to get them. And I mentioned all the image classes in SFE require spatial extent, and we use the ext function. So this is actually it's just to keep it consistent with Terra. So this will get the spatial extent of xmin, xmax, ymin, and ymax. In Voyager, the plot image function can be used to plot the image alone without the other geometries. Or depending on the class of the image, like here it's from Terra, so you can also use Terra's plotting function if the image is already Terra. And the image, so plot spatial feature is the main function to plot the cell level data in Voyager, and you can so here you can, by populating the image ID argument, you can plot the image behind the cell level data. And actually the same can be done for plot geometry or plot local result. Just can be done for any function that plots the geometries. So what's special about Terra here? So I mentioned there are 3 classes. So that's because there are advantages and disadvantages associated with each tradition. And Terra is from the geospatial tradition. The advantage is that we can use vector geometries to like from SF to extract data from raster. Like here, I will use the extract function from Terra to extract like polity intensity from like behind each cell segmentation polygon. But the disadvantage of there are disadvantages with Terra in that so we cannot apply like image processing techniques. Like image processing is like a whole other tradition. In R, actually I usually use Python for image processing, but in R it's not that great. But in R, the main package, like in Bioconductor, the main package for image processing is EB-image. So here we can convert the spat raster to like XD image. So that's the XD image class inherits from the image class from EB-image. So it's actually all it does is to add the spatial extent to align the image to the geometries. So here I use the coarse function to XD image to convert that to the EB-image required class, and the EB-image has a nice little interactive widget to plot it. So with EB-image, we can apply more methods from the image processing tradition. Like here, I'm applying Otsu thresholding, so that's a way to automatically detect the threshold for like segmentation. So here, I just want to see like I just want to distinguish the tissue from like empty space here with Otsu thresholding. And we see here that, we see here that like there's a small bit and there's a hole, and from the image processing tradition, we can do like opening operation to, opening can remove the small bits and closing can fill in the holes. That's what it would look like. So here it's a mask, and it's a dense matrix. And so in case you actually this can be like from Terra, I can convert it back into SF polygon. So we can store the polygon, which is to save a lot of memory and disk space as opposed to the dense matrix in the mask. And here, because actually in the mask, although I apply the image processing methods from EB-image, it actually still has the spatial extent. So we still know where it is in the spatial coordinate system. And so here I convert it back to spat raster and then like I use this is just a Terra function to convert the mask into polygon. Yeah, and that's what the polygon looks like. And I will add a polygon back to the SFE object for like bookkeeping. So yeah, that's what it will look like. Any questions? Oh yes. No. Okay, I can continue. Yeah. So another type of image in SFE is called BioFormats image, and so that's how I deal with like OME-TIFF, like basically anything that can be read by the BioFormats Java library. And so there's the R BioFormats package. Actually, the reason why I use EB-image is that R BioFormats is written by the same person. So if I use R BioFormats to read the image into memory, it's going to be EB-image. And so here for Xenium, we have OME-TIFF files, and actually the OME-TIFF files have like a kind of data compression that Terra doesn't support. So that's like my original reason for writing BioFormats image. And here, we see here we have an image with 4 channels, and it's BioFormats image. And I can use the get image getter to get this image. So here it prints out the summary of the dimensions and like 4 channels here. For BioFormats image, actually, this image can often in Xenium, these images are pretty large, and those are actually pyramids with multiple resolutions. And usually I don't want to read the highest resolution into R, but here actually for this class, the image is not read into memory at all. I merely read the metadata from the OME-TIFF file, and only the metadata is read into R. So the metadata in memory will have the spatial extent. So as we will see later, we'll also store the information about affine transformations if we perform affine transformations on the data. So here I have 4 channels, but we only have RGB, and plot image can let you select up to 3 channels to plot as RGB channels. This is what it looks like. But because it's RGB, it's not colorblind friendly, and so we can also plot the channels separately. And yeah, so that's using that's in grayscale, or we can use different palette if only plotting one channel. Like here we use a brightest. Like multiple channel images can also be plotted behind the images. And I mentioned it's a pyramid, and so in plot spatial feature and all the other plotting function with dealing with images in Voyager, it will. So actually, there's an argument here in plot spatial feature that will limit the number of pixels that can be read into memory, and then this function will will get the metadata and will automatically determine the highest necessary resolution to from the pyramid to be read. So usually, it doesn't, unless you're really zooming in, you're not reading the full resolution image. And even if you are here, like I have supplied a bounding box, and it will only read that small part of the image necessary. So usually you don't need to read the whole thing to memory. And in order to read BioFormats image into memory, you use the 2x image coercion function. Yeah, so this has a method for spat raster like from Terra, and also has a method for BioFormats image. And here, because I read the second resolution, so lower resolution into memory, and it's already yeah, and it can be plotted with the widget in EB image. So okay, I'm kind of short in time. So we can perform like operations on SFE objects. I already covered the non-spatial. Like yeah, you can also like do C-bind just like for matrices. That's for non-spatial. And this slide is a summary of the spatial operations that can be done. And I already mentioned the vector raster interaction. But we can also use a bounding box or any kind of polygon to crop the SFE object. So this way you can only get like a small part of it, or like it doesn't even have to be square. It can be a circle or anything. And we can also perform affine transformations and spatial aggregation and so on. And affine transformations will help with aligning like multimodal data from adjacent sections. Like in this plot here, I used an affine transformation to align to align a Visium dataset to a MALDI-MSI metabolomics dataset. And so, yeah, like for the spatial transformation, like from SF, for any SF data frame, you can get a bounding box. But for SFE, you can also get a bounding box, and because SFE you can have any number of geometries for like row, column, or annotation. The bbox method for SFE here really we'll find the largest bounding box for like all geometries, and optionally you can also include images, but although by default it does not include images. For cropping, there's the crop function. So the first argument will be the SFE object, and the second argument you can put a bounding box. Like it can be, like in this case, the bounding box is just a numeric vector with names. But it can also be an SF data. It can be like an SF data frame or like a SFC. I think the geometry can be any kind of geometry. So here I'm cropping the SFE object with this bounding box. It's quite a small one, and so after cropping, initially we have here we have like 5,700 something cells, and after cropping we only have about 200 cells left. And to plot the cropped SFE, like I just supplied the SFE cropped, and here I did not supply the bounding box, and that's what it looks like. Affine transformation. So there many methods for their specific transformations are named like mirroring vertically or just flipping vertically or horizontally, or we can also scale or rotate or transpose and so on. So those are named, and you can call the functions by name. Like here's mirror. And in this example, we will mirror the SFE Xenium dataset vertically and plot it. Yeah, so earlier, that dataset will looks like this. That's what the image originally looks like. You see the blood vessel like running this way, but after flipping it, after mirroring it, it will go it went the other way. And note that actually I also plot the image behind it, and so after flipping, actually all the geometries and the image those are all also flipped, and they will remain aligned. It's just like in the map. Just like in the map, like you can scale it, and you can also rotate it, but everything will still remain aligned. But more generally, say for a purpose of aligning different sections, you can supply a transformation matrix. So it can be a 2x2 matrix for linear transformation, and another vector for the translation. So here it's for this example, although there is a rotate function, I just put a rotation matrix to rotate it by 30 degrees, and this is what it looks like after the rotation. So again, like everything would remain aligned. But then what happened to the image after the rotation? So after, actually, when I'm not doing the plotting, it's BioFormats image are, the BioFormats image is still like not loaded into memory. It's still BioFormats image, so it still only has the metadata in memory. And but the rotation is done when I just store the transformation matrix in the metadata here. And so when I plot this, the plotting function just loads a low resolution version of the image, and then applies the affine transformation on the fly when plotting. So another kind of spatial operation can be spatial aggregation. Here it's worth mentioning the SE raster package, which also which uses aggregation by a lower resolution spatial bin when you have like lots of cells. And after spatial binning, the SE raster paper showed that it really drastically improved the speed of like finding spatially variable genes. So here we can, SCP also has its independent implementation of the spatial binning. It can be done at the level of transcript spots. You can do that like even without calling the read Xenium function first. So here I'm just supplying the output directory, and just say it's Xenium, and then it will just read in the file with the X and Y coordinates of the transcripts, and then here from cell size. So that's I will aggregate those with bins. Square equals false, so it's hexagonal bins, and I aggregate the transcripts with the diameter bins with diameter of 20 microns. And so here by aggregation, I just mean like how many transcripts there are in each of the bin cells. So after aggregation, I get an SFE object, and the call geometry would just be the bins. Then we can plot the total number of transcripts in each bin, and that's what it looks like. Meanwhile, there's an aggregate method for SFE, and so you can. So when you already have an SFE object and you already have the row geometry for transcript spots, you can so you can also like here by cell size, so like by default square equals true. So here I'm aggregating the transcript spots from the SFE object into a square grid with each side is 50 microns. And this is what it looks like. So now I can get a little bit into Voyager, but that's actually so you can see you can go to the last year's version of this workshop, and you can also go to the documentation website if you want to look at more details about the ESDA methods. Yeah, like there will be a bit of intro about the math, but I will not cover that today. So the philosophy of Voyager is that so although here we see that the spatial transcriptomics really took off after like 2019, but there is a glorious tradition in spatial data analysis, mostly coming from geography that are many decades old. And Voyager basically brings a lot of like decades of research in spatial analysis into into spatial omics through SFE. I'm now the first person to bring geospatial tradition to spatial omics. So this slide lists a lot of the existing methods that already do that, but they will focus on very specific tasks. While so like in exploratory data analysis, it's like in John Turkey's own word, it is important to understand what you can do before you learn to measure how well you have simply done it. And the other packages today focus on specific tasks and try to do it well. So that's how well you have seemed to done it. But Voyager will focus on the step before that about the what you can do part. So this is why it has a uniform user interface that can give access to many different ESDA methods from the geospatial tradition to allow you to explore around. Like a lot of them just a quick and dirty way for you to ask questions before you narrow down like which questions are worth further investigating. And for the further investigations, you can use like other methods from spatial omics or geospatial tradition. And here, one of the quick and dirty things I'm trying to do is to use the spatial aggregation to investigate spatial autocorrelation at different scales. So actually another way to do the spatial aggregation. So just now we supplied like the data directory, but here in order not to read in the same file, like not to repeat the read operation like over and over again, like I can read this into memory first, and then I create like a sequence of different bin sizes. And then here I apply the spatial aggregation like over different bin sizes. Right now, yeah, what is spatial autocorrelation? I mentioned it. It's a big topic in ESDA. Basically means it's like everything's related to everything else, but near things are more related than distant things. For instance, there can be positive spatial autocorrelation, like nearby things tend to be more similar, just like in weather. But there can also be negative spatial autocorrelation, which is very much neglected because there are way more examples of positive spatial autocorrelation than negative spatial autocorrelation. For negative spatial autocorrelation, the nearby things tend to be more different. Like on the chessboard, the neighbors must be different. The most common measure of spatial autocorrelation is called MALDI-MSI. I would not go into the math in detail here, but it's basically it's a little bit similar to Pearson correlation, but not the same. It's just like Pearson correlation. It usually ranges between -1 and 1, except in like pathological cases. And if you have a positive number, it means that you have positive spatial autocorrelation. If you have a negative number, you have it indicates negative spatial autocorrelation. So here so this is still running. Here, yeah, it just finished. So here SFE, so that's a list of SFE objects. Like aggregate, it just aggregates the transcripts at different resolutions, and what I'm trying to do is to compute Moran's-I at for like all the genes at different spatial resolutions.
Quick question.
Yes.
Oh, I think this was from a little bit ago, but when they're talking about the aggregation, Vineeth wanted to know if you're aggregating multiple Xenium samples.
Actually, that can still work. Here's here's like a subset of it. Like it's the same for the full thing. Yeah, although like I haven't done the benchmarking yet. I could do some optimization, but like in software engineering, there's a saying that like premature optimization is the source of all evil. So I'll do that a bit later and to make sure it's fast for the full data set. Yeah, so in order to compute Moran's I will also make the spatial neighborhood graph. Here I make two different types, like one's rook, one's queen. So rook is like on a chessboard. You only do like a horizontal and vertical. So like the neighbors must touch by at least an edge. Queens, like you can also do diagonal. So it will also count if they only touch by a point. So here it's just a sort of to show the implications of using different spatial neighborhood graphs. Yeah, I have the bins here. That's what the smallest one looks like, and that's what the largest one looks like. Yeah, here I compute Moran's I for just total count. So for like call data, we can use the call feature data getter to get the spatial analysis results for like call data. Here just a bit of data wrangling to make the plot of how this Moran's I will vary by spatial scale, and here we see that in this plot. So at a smaller scale, it's pretty high, but then it decreases for intermediate scales. But before it gets larger again for a larger scale. Actually, in Voyager, there are already methods for to explore the length scales of spatial autocorrelation, like in parallelogram. But at least in SP-DEP's implementation, which Voyager wraps around, the parallelogram is pretty slow to compute. So like spatial binning could be like an alternative that could be faster to compute. Also, like, why would this be interesting for? Actually, can when you have the spatial neighborhood graph at single cell resolution, like in Xenium and MERFISH, and or like it actually means different thing from in Visium, where each spot has many cells. Because at different scales there can be different spatial phenomena. Like here in geographical space, the spatial phenomena at the level of like wildflowers will be very different from that on the level of landscapes. And actually, in one of the this examples in the first MERFISH vignette in the Voyager documentation website. So here at the single cell level, you can have a negative Moran's I, but after spatial binning, it will flip sign. So that's an example to show that it can be interesting to investigate this. So we can. So that was for transcript level, which I personally think it would be more appropriate. But we can. SC raster will do the aggregation at the cell level, and we can also do that here with the aggregate function, which is popularly the call geometry name instead of row geometry name. So at cell level, we can have we already have the gene expression data, and we can use different functions to aggregate those. Like we can use sum or mean or like any other function. Yeah, it's just a sum and mean. Actually, behind the scene, I'm using matrix multiplication to compute the sum and mean. But for the other functions, I have to loop through the like each individual spatial bin. But that can be parallelized, but that's still slower than the matrix multiplication. And here I got sum, mean, and the median. Actually, choosing the choice of the aggregation function, the summary function, like sum as opposed to mean, will have implications to further analysis. Like here, they actually look quite different. Yes, I think you can read that later on the discussion of this. But yeah, I'm almost out of time. So last one is the split. So here we can, so that's the opposite of aggregate. And so here the special part is that you can use a geometry to split a spatial feature experiment object. So here, say I use this one, these polygons, and the cells in this part, in this piece will become one SFE object, and cells in the other piece will be a separate SFE object. So it's done by the split by call function. It's again uses the geometric operations, and Voyager we can also like put them back together into like just different samples and plot them, and that's what after splitting what it looks like. And the spatial neighborhood graphs are also split, and so here we can. This is just like an artificial splitting of samples, but splitting can be more relevant in real data set when say you have a TMA, tissue microarray. You can so where you have like small pieces of tissue from different patients mounted on the same slide. And here so for the multiple samples, like it can be from like case and control. We can also like compare the Moran-MSI from those. Although here it's I'm running Moran-MSI for all the genes, and for delayed array, it can take a while, but here's an example of the comparison. So I have a Visium dataset from a collaborator from UC Irvine, and I have case and control. So the case is the knockout, and I was asking, so which genes are spatially structured in one of those conditions, but not the other. And it turns out there's some those will encode for the those genes will encode for the immunoglobulin. So probably like an immune foci for so that looks interesting, but this will point to again the ESDA methods are I would say like it's not like a scale, like to paraphrase my advisor Lior, it's not like a scale where whose physics is well known and you press a button you get the results, but in bioinformatics, the data analysis methods, I think it's actually more like an art, more like a palette, or more like even Art Studio. It doesn't write the code for you. It doesn't make the art for you. You still have to think about it. It's not like a scale. So here there are other issues when it comes to like experimental design. For when I did the same comparison for the adjacent sections, I also get like those genes are also have much higher Moran-MSI in this section compared to this one. They're actually 0 sections. Just because like this section catches more of that immune foci compared to the other one, but what if like in this section is just unlucky to miss it? So basically there are many open questions. This is done computing, and for gene expression, the results are stored in raw data, and so we can plot those. Just some data wrangling. Because this is the same piece of tissue, the Moran-MSI is usually consistent, but we do see there are some. So each point is a gene, and we see like there's for some genes the Moran-MSI is quite different between two pieces. What does it mean? Is it even significant? So that's an open question, and in the text I have more discussion about it.
Quick, we're just a little over time. Do you have a quick question?
Depends. Lambda, a great demonstration. I was wondering, you mentioned Seurat and that you provide some coercion functionality there. There's another competitor in that space that I think you're familiar with, Giotto. And I was wondering, have you looked at all a little bit into quantitative comparisons between the three, Giotto, Seurat, your framework?
Actually, yes, it's I think it's in the Voyager preprint. It's a bit old. I compare I compare PCA, DE, and actually also Moran-MSI. I also compare with SquidPy for Moran-MSI. Also there's a student in my lab at Caltech, Joe Rich. I think he's going to give a presentation about a comparison between Seurat and ScanPy, but although that's not for spatial, but I did that for Moran-MSI though. For at least that's what what SquidPy, Giotto, and Seurat and Voyager have in common.
I think very interesting would be to look at the geometric operations, right? Geometric overlaps, spatial overlaps, and I think
but I mean,
think a little bit about the backends, right?
Yeah,
uses SP, you using SF, Giotto
Yeah,
uses
it's
Sarah, and I think there you can work out some clear benefits of one over.
Yeah, I think that's not always supported by. It's not supported by all those packages. So like in some cases, it's not applicable. So I think yeah, it could be I think it could be worthwhile to make sure that things are like comparable across different platforms. Yeah, I also hear like I use geos actually behind SF. It's actually geos and GDAL, so it can depend on the backend. But if they use the same backend, then I would expect the same results.
There's no other questions online other than to please share the URL of those presentation slides as well. And so it says if once ever you get them, put them up. Also, you can maybe post it onto the Slack.
Yeah. So I'll put the slides in my GitHub repo and push it, and yeah, I think I'm going to post the URL to the rendered version of the workshop now. Here.
This has been an amazing demonstration of a lot of coding and functionality. So everybody, thank you. Join me in thanking Lambda for a wonderful workshop.
All right, thank you.
Okay, and I guess
the ESDA part, like also when it comes to the comparison of Moran's, I think those are all open questions, and here I'm just only showing like a quick and dirty thing to get into like something interesting, but I don't know if it's significant. So there will still need